Welcome to the lesson Using Bioinformatic Tools to Validate Your AI ML Results. To set the stage, imagine you've just run a machine learning pipeline which has identified some genes most predictive of a phenotype. Now you want to determine what is the biological function of the genes you found. If the genes are involved in physiological processes which are known to be associated with your phenotype, then that serves to help validate your results. Moreover, knowing which genes are implicated in these deleterious physiological processes provides a molecular target for diagnosis, monitoring, and potential therapy. The objectives of this lesson are to perform differential gene expression analysis, perform pathway enrichment analysis, and perform gene set enrichment analysis. In this first section, we describe how to perform differential gene expression analysis. Recall the central dogma of biology. The so-called central dogma of molecular biology defines the flow of information from DNA to proteins through an intermediate form called RNA. DNA itself is a double-stranded molecule residing inside the nucleus. When a protein needs to be expressed, the gene or genes that comprise that protein are transcribed into RNA inside the nucleus. RNA are single-stranded molecules with a slightly different molecular composition than DNA. The RNA fragments are further processed and exported out of the nucleus and into the cytoplasm, where they are translated into proteins, one amino acid at a time. DNA also undergoes replication during cell division. RNA sequencing is the process of isolating, amplifying, and counting these RNA transcripts to see which genes are active in a cell or tissue at a particular point in time. Recall RNA-seq. RNA sequencing has two primary components: a wet lab component where chemical reactions are used to extract, purify, amplify, and sequence RNA fragments, and a dry lab component where those RNA fragments are quantified and analyzed to ultimately provide biological insights. Recall RNA-seq prepared data. After the RNA transcripts have been prepared, their counts are stored in a table such as what is shown in the slide. In this slide, ground control sample GC9's gene 142 has 357 transcripts, whereas flight F20's count for the same gene is 389. Is the collective difference between all the ground control samples and all the flight samples statistically significantly different for this gene or any other gene? That's the question that differential gene expression analysis answers. Differential gene expression analysis is a statistical experiment scientists perform on RNA-seq count data. There are two groups of interest, say A and B, one of which is the control group, to which no changes were made, and one of which is the experimental group, to which some change was made. The experimental change can be anything, such as treated with a drug versus untreated, or other so-called changes like male versus female, or old versus young, strain 1 versus strain 2, smoker versus non-smoker, etc. In our case, the experimental change is whether the organism was flown in space, i.e. spaceflight, or not, i.e. ground control. In our case, we wish to know if some genes are over or underexpressed as compared to the baseline, and any such genes we will call differentially expressed. The biological explanation for differential expression is that an organism is trying to compensate for the experimental change in order to maintain homeostasis within the cells, organs, and systems. How do you visualize differential gene expression? The most intuitive way to visualize differential gene expression is to create what is called a volcano plot. A volcano plot, so named because it looks like an erupting volcano, has two axes. The horizontal axis represents fold change in log scale, and the vertical axis represents the p-value in negative log scale. Fold change on the horizontal axis is the ratio of the group A expression to the group B expression in log scale. So for example, if some gene has a mean count of 128 in group A and 8 in group B, then the ratio is 128 to 8, or 16, and the log base 2 of 16 is 4. A log2 fold change of 0 is when the mean count of group A is equal to the mean count of group B. And a negative log2 fold change occurs when the when the group A expression is less than the group B expression, because the log of a number less than 1 is negative. The further to the right is the higher the overexpression, and the further to the left is the lower the underexpression. The p-value, which is on the vertical axis, represents the overall statistical significance of that expression value relative to all the other gene expression counts. We use the negative log of the p-value because the p-value is a number between 0 and 1. So if the log of a number between 0 and 1 is negative, and we want our plot to sit in the first quadrant of the plane, the negative is a positive. The higher the value on the vertical axis, the more statistically significant the fold change. In all, then, the further up and to the right or the further up and to the left are the most significantly differentially expressed genes. How does it work? Differential gene expression analysis frames the question, is the observed gene count A0 significantly different than the mean A hat as a hypothesis test? The null hypothesis H0 states that they are not statistically different, and the alternative hypothesis H1 states that they are statistically significantly different. In general, we don't care if A0 is significantly greater than or significantly less than A hat, so we measure the unsigned distance. This is called a two-sided test. First, we start by dividing all the log fold changes by their standard deviation to get a standard normal distribution, which is what you see in the picture. In this graph, A hat represents the mean of this distribution, and A0 is the observed value of a gene count. The question is, how different is the count A0 from the mean A hat? As you can see, there are three different ways to measure that: the likelihood ratio test, the score test, and the Wald test. Differential gene expression analysis works differently depending on the software package implementation. The software package we will discuss in the next few slides is called DESeq2, and it provides both the log likelihood ratio test and the Wald test. Let's discuss the Wald test, which is the default in DESeq2. The Wald test calculates the probability of observing a z statistic at least as extreme as the observed value A0 randomly. If the p-value is sufficiently small, i.e. less than 0.05, we reject the null hypothesis, which means we accept the alternative hypothesis that the value A0 is significantly different. Which tools are available to perform differential gene expression analysis? There are several tools available, as shown in the slide. The tool we will use in our lab is the Python implementation of DESeq2 called PyDESeq2. The other three tools shown in the slide, Lemma, EdgeR, and DESeq2, are supported by an open source project called Bioconductor and are all implemented in R. Here's an example of performing differential gene expression analysis using DESeq2. In the first part, we show on the left a table that associates for each sample F9, F11, and so on up to GC20, a condition value of 0, meaning ground control in this case, or 1, meaning spaceflight. In part 2, we have our table of gene expression in which each row represents a sample. For example, the first row is for F9, and each column represents a gene. For example, the first column is GNAI3. Each cell is the count of RNA transcripts for that gene for that sample. So for example, 226 is the number of RNA transcripts measured for sample F9 for gene GNAI3. In part 3, we show the sequence of Python statements to execute the DESeq2 experiment, which ultimately generates a list of statistically significantly differentially expressed genes. Recall that a differentially expressed gene may be overexpressed, meaning that the experiment group's mean expression is significantly higher than expected, or underexpressed, meaning the experiment group's mean expression is significantly lower than expected. Finally, in part 4, we show the list of those 10 genes. Now the question is, what do we do with this list of genes? But before we get there, I just want to point out a few things. For example, in step 6 here in part 3, we're only considering the results of genes whose mean is at least 10. And then in line 7, we're looking at the significant results. And by significant results, we mean your adjusted p-value is less than 0.05, and your log 2 fold change, the absolute value of your log 2 fold change is greater than 1. And that's how we get our gene list in this case. Now, what do you do with this list of genes? Well, first, you can look up the genes online one by one. And there's lots of tools to do that. There's a tool called NCBI, which provides a portal for you to look up genes one at a time for multiple different species, or organisms rather. Or you can do what's called pathway enrichment analysis, and there's a tool that we call Shiny Go that we're going to be looking at in this lesson. You can do what's called gene set enrichment analysis, and there's a tool called MSigDB for that, which we will also be looking at in this lesson. And then finally, you can also use them in conjunction with your AIML pipeline. So here first is looking up this gene Cal4A1 in that NCBI tool. So you can see it gives you the official full name, whether it's protein coding or not, what organism it's coming from, other other names, also known as, and then a summary of the function of this gene. And then this is just the first part of this page. If you scroll down in this page, you get to the phenotypes. And in this particular list of phenotypes includes one of which has to do with the retina and the other with cataracts, and both of which are consistent with our observed phenotype of space-associated neuroocular syndrome. How can I use differential gene expression analysis and those 10 genes with my AIML pipeline? Well, there are a few ways you can do that. First, you can compare the results of your model to the gene list you obtained from differential gene expression. Is there any overlap? If there is, what does that tell you about your findings with your modeling? If there is no overlap, what does that tell you about your AIML findings? A second way to use differential gene expression analysis results with AIML is to use differential gene expression to reduce the number of genes under consideration in your modeling. So recall, RNA-seq experiments typically have 20,000 or more genes, and having only a few ground control samples and spaceflight samples is not sufficient to determine the effect for each gene for each group. Dimensionality reduction is a method in AI and ML to reduce the number of features, and this is one method of dimensionality reduction. And conversely, you can use, if you want to provide your results from differential gene expression, but first want to filter out some genes to reduce dimensionality, you can use your AIML model to do that reducing first, and then get your results out of differential gene expression. So in this section, we will perform pathway enrichment analysis, which is a particular type of gene set enrichment analysis. This is one technique which will answer the question, now that I have a list of interesting genes, what do I do? So what is pathway enrichment analysis? In pathway enrichment analysis, the goal is to determine whether a predefined biological pathway, for example, a signaling cascade or a metabolic pathway, is overrepresented in a list of genes of interest compared to what would be expected by chance. This analysis helps to define which pathways are potentially involved in the biological process under investigation. Pathway databases like KEGG and Reactome provide predefined pathways that can be used for this analysis. In this slide, we see that our list of 10 differentially expressed genes, or DEGs, EPSBL1, IRF7, and so on. We also provide the list of background genes, which is the original set of approximately 20,000 genes we used in the RNA-seq experiment. In the green box in the middle, we see a partial list of the gene sets, which represent different biological pathways, such as the insulin pathway, the fatty acid biosynthesis pathway, and so on. Each of these pathways has a set of genes which creates proteins that are involved in that process. Pathway enrichment analysis then determines if your list of differentially expressed genes is enriched in any of the gene sets to find overrepresented ones. These overrepresented pathways then are putative processes that are either switched on, upregulated, or switched off, downregulated in your samples. How does pathway enrichment analysis work? Pathway enrichment analysis runs a Fisher's exact test from a 2x2 contingency table to find whether the genes in the gene list are statistically significantly enriched in a pathway or not. In other words, what are the chances of drawing those genes given that they are in those pathways as opposed to drawing them completely by chance? The rows of a 2x2 contingency table define two Boolean states, in the pathway, not in the pathway, as columns, and two other Boolean states, in the gene list, not in the gene list, as columns. Sorry, the first one was for rows. To calculate the value, just plug in the values for M, which is the total number of genes in the pathway, for X, the total number of genes in the gene list that are in the pathway, for K, the number of genes in the gene list, and for T, the total number of genes in and not in the pathway. Of course, these are very large numbers, and there are tools online such as GraphPad that we can use to plug into a 2x2 contingency table to calculate the p-value for Fisher's exact test. So Fisher's exact test for this particular 2x2 contingency table, representing the 5 genes in the list, 4 of which are in the pathway and 1 isn't. The 500 genes total in the pathway and 4,500 total genes not in the pathway. A p-value of 0.0005 is extremely significant. We would then conclude that this list of genes, RRP6, MRD1, and so on, are significantly enriched in this particular pathway, whichever one it is. Which tools are available to perform pathway enrichment analysis? Here's a subset of the commonly used tools to perform pathway enrichment analysis. Note that EnrichR and David do both gene set enrichment analysis and pathway enrichment analysis. In the hands-on activity, we'll be using ShinyGo. Here's an example of performing pathway enrichment analysis using ShinyGo. The slide screenshot shows an example of this online tool. In 1, you select the species, in our case it's mus musculus or mouse. In 2, you paste the list of genes you found as interesting, those 10 genes that we found as differentially expressed. In 3, you paste the list of background genes, which is the list of all the genes, those 20,000 genes that you used in your experiment. In 4, you select the relevant pathway database. It defaults to the KEGG database. And when you click the Submit button, you'll get a table of pathways which are statistically significantly enriched by your list of genes relative to the background genes. In this example, there are a number of pathways seemingly unrelated to our spaceflight-associated neuroocular syndrome phenotype based on the name. However, if you look more closely at each of these pathways, you'll see that the 2 genes which enrich those pathways are Cal4A1 and Cal4A2 genes, both of which are responsible for making collagen, which happens to be the most abundant protein in the human body. Because our gene list comes from an RNA-seq experiment on retina tissue, we would then ask, what is the function of collagen in the retina? For that, you can use your favorite internet search engine to look at research papers which have explored the function of collagen in the retina, and you would find that it's primarily responsible for maintaining intraocular pressure, which is directly related to our spaceflight-associated neuroocular syndrome phenotype. How can I use pathway enrichment analysis with AI and ML? The graphic in the slides shows how you might do that in biomedical research in general. The AI/ML model will presumably give us as its output a list of genes which are associated with a response or phenotype. Taking this list of genes and plugging them as input into a pathway enrichment tool, such as ShinyGo, will provide a list of pathways which are significantly enriched by those genes. The databases of pathways which are consulted are typically configurable by the tools we saw, so you might choose to restrict your search only to the databases you think are relevant to your phenotype. Once you get those functionally enriched pathways, you can begin exploring the scientific literature to see what effect those genes have in those pathways with respect to the phenotype you're observing. In this section, we perform gene set enrichment analysis. This is a second technique which will answer the question, now that I have a list of differentially expressed genes from my AI/ML model, what do I do? What is gene set enrichment analysis? Gene set enrichment analysis is a computational method used to determine whether a predefined set of genes shows statistically significant concordant differences between 2 biological states, i.e. phenotypes or conditions. It was originally developed at the Broad Institute And gene set enrichment analysis is particularly useful for interpreting genome-wide expression data like that from an RNA-seq experiment. The primary goal of gene set enrichment analysis is to identify whether the members of gene sets, which are groups of genes that share common biological function, chromosomal location, or regulation, are randomly distributed across a ranked list of genes or primarily found at the top or bottom. This helps researchers understand whether particular functions or pathways are upregulated or downregulated in a given condition. How does it work? Here's a simple breakdown of how this generally works. First, we rank the genes. The genes are ranked based on their correlation with a specific phenotype or condition, A versus B. This might involve metrics like the difference in expression levels between the two conditions or correlation with phenotypic traits. Two, we define your gene sets. Gene sets are defined based on prior biological knowledge, such as pathway databases like the KEGG pathway database or the Reactome or BioCarta. They're also based on gene ontology terms or other functional classifications. Then we calculate an enrichment score, ES. So gene set enrichment analysis calculates this enrichment score to reflect the degree to which a set of genes is overrepresented at the extremes, either the top or the bottom of the entire ranked list of genes. The score is essentially a running sum statistic computed by walking down the list of genes, increasing the running sum when encountering a gene in the gene set, and decreasing it otherwise. So then basically we take a walk from the left to right going down those genes, and you can see that the ES of that gene set is the distance between 0 and the highest peak, basically, and that's going to be your enrichment score. There's some other things we need to do, like we need to normalize because not all the gene set lists have the same length. We also have to do multiple hypothesis testament adjustment because we're asking the same question 20,000 times. So there's a number of adjustments here, but this is the primary way we get the enrichment score. Which tools are available to perform gene set enrichment analysis? Well, here's a subset of the commonly used tools: EnrichR, MSigDB, and David. MSigDB is the one we're going to be using in our hands-on. And here's an example of performing gene set enrichment analysis using MSigDB. So first, in 1, you paste your list of genes into the text area. Second, you select the species, either human, mouse, or rat, in that drop-down list. Third, you select the gene sets you wish to explore. Here we've selected C5, which represents all the ontology gene sets. After clicking the Compute Overlaps button in the 4th step, you'll be shown the list of gene sets in 5, which were statistically significantly enriched by the gene list you provided. The gene sets are accompanied by a brief description, the number of genes in the overlap, the FDR-adjusted Q value, showing how statistically significant that overlap is. And recall, anything below 0.05 is generally considered statistically significant in scientific research. Now, looking more closely at the gene set names, you'll notice that most of them have to do with abnormalities in eye function. Take the first one, for example, called HP abnormal lens morphology. If I click that link, the MSigDB tool shows more information about that gene ontology term, as well as the genes that comprise the gene set. This phenotype is consistent with our hypothesis that spaceflight has a negative impact on neuroocular function. Moreover, these gene sets inform us as to how exactly the eye may have been impacted, giving us clues to pursue for further research into spaceflight-associated neuroocular syndrome. Now, how can I use gene set enrichment with AI/ML? Well, do your result genes from your modeling enrich gene sets that relate to your phenotype? And that's it. That brings us to the end of the lecture in which we discuss performing differential gene expression analysis, pathway enrichment analysis, and gene set enrichment analysis. In the hands-on activity, you'll have the opportunity to experience using each of these tools and interpreting their results.